There are four types of macromolecules made by cells only: carbohydrates, lipids, proteins, and nucleic acids. These types of macromolecules are organic compounds because they all contain carbon. Carbohydrates are for quick energy and they have some structural use. Lipids are for stored energy. They're for long-term energy use. And we do have some hormones that are lipid-based. And lipids make up our cell membranes. Proteins are enzymes, and most of the structure of our body are proteins. And really, proteins, there are too many functions to list, but when we get to the protein sections, I do list a lot of proteins. And for the nucleic acids are DNA, that's deoxyribonucleic acid, and ribonucleic acid, RNA. Now carbon, which is the basis for organic compounds, if you have to have carbon to be classified as an organic compound, is electroneutral, meaning that it does not ever swipe electrons from other atoms. It's electroneutral because it contains four valence shell that should say electrons. Sorry about that. That's a big typo right there. It always has four valence shell electrons. They're never neutrons in the valence shell. It always has four valence shell electrons. Carbon always participates in covalent bonding. When you think of C for carbon, think of C for covalent bonding. It never ever participates in ionic bonding. Never, under any circumstances. Carbon always participates in four covalent bonds. Now, before we get into our types of organic molecules, our macromolecules, we need to go over dehydration synthesis and hydrolysis. For dehydration synthesis, what we're going to do is we're going to build something bigger by the removal of water. So here we have monomers, and we're going to build a larger unit called a polymer. So we're taking two smaller something or others, and we're building a larger unit. And when we do that, we're going to take a hydroxyl unit off of one monomer and a hydrogen ion off another, and we're going to build a water molecule. So that's why it's called dehydration. That's the dehydration part of this dehydration synthesis. And the synthesis part is where we're building something. So in dehydration synthesis, we take a monomer, two monomers, and we add them together to build a polymer. So we're going in this direction. Here are our reactants, and this is our product. In hydrolysis, we have the reverse. Our reactant is something larger, and we're going to break it down into the products, which are something smaller. So here we have, in hydrolysis, we're taking a water molecule. It requires water, and we're going to literally wedge that water molecule up in here, and we're going to break this covalent bond, and we're going to break it into two smaller units, two monomers. So here we're having the addition of a water molecule. We're taking this water molecule and we're applying it to this monomer. We're adding an OH over here and an H here. So we're breaking this polymer down into its monomers. That's hydrolysis, literally breaking with water, hydrolysis. So dehydration synthesis is building something larger, and we're releasing water into the environment. We're dehydrating these two monomers, and hydrolysis is splitting with water, but it requires water. So think of this as like in digestion, it requires water to digest something bigger. If this were protein, we'd be breaking protein, which would be like the polymer, down into its individual amino acids, which would be the monomers. So here's an example reaction. In this reaction, if we're building something bigger, if we're going in this direction here, this would be dehydration synthesis. And we have two little monosaccharides. We have glucose as a monosaccharide, and fructose as a monosaccharide. And we're going to take this little hydroxyl of glucose and this hydrogen off fructose, and we're going to make a water molecule. And we're going to release that into the environment. That's the dehydration part of the synthesis. And we're going to form this disaccharide, sucrose. If we go in the opposite direction, if we take the sucrose and we put it into our mouth and we digest it, we're going to break it with water. We're going to take a water molecule and we're going to bust this covalent bond. We're going to take that water molecule and we're going to put it in here, and this glucose molecule is going to take the hydroxyl portion of it, and the hydrogen is going to go onto the fructose, and we're going to end up with our individual monosaccharides. So water is going to be consumed in this process if we're going in this direction, the hydrolysis direction, the digestion. So we have hydrolysis and dehydration synthesis. So now we're going to actually get into our first organic compound, carbohydrates. Carbohydrates are made of carbon, hydrogen, and oxygen, and our simplest carbohydrate is going to be the monosaccharides, and we call those the simple sugars. Mono means one, and sac means sugar, so these mean one sugars. Now, our disaccharides mean two sugars, and polysaccharide means many sugars. So we call these our complex carbohydrates or our many sugars. If we take a monosaccharide and hook it onto another monosaccharide, we get a disaccharide. If we put many monosaccharides together, we get polysaccharides. So we call this the monomer and the polysaccharide the polymer. So down here we have monosaccharides, disaccharides, and polysaccharides. Glucose and fructose are examples of monosaccharides. This is sucrose, is an example of a disaccharide, and starch, and cellulose, and glycogen are examples of polysaccharides or the complex carbohydrates. If we look at the monosaccharides, monosaccharides consist of carbohydrates, oxygen, and hydrogen, and those atoms are usually in a 1 to 2 to 1 ratio. So if we look down here at the molecular formula for glucose, we see that this is a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. We have 6 to 12 to 6. And here for ribose, here's the formula, 5 to 10 to 5. Down here, this is a 1 to 2 to 1 ratio. That should be an O, so sorry about that. There's a typo. And now also what we want to know about our monosaccharides is they're considered pentose or hexose usually. Pentose means we have 5 carbon atoms, and hexose means we have 6 carbon atoms. And these monosaccharides are very soluble. If you put them in water, they easily dissolve. Usually they're sticky, very sticky. Glucose is our blood sugar. Ribose is the sugar that we find in RNA. Fructose is our monosaccharide that we find in fruit. And deoxyribose is our monosaccharide that we find in our DNA. So those are our monosaccharides that we want you to know examples of. Our disaccharides are two monosaccharides that you link together covalently. Those are strong bonds. Now remember, all of the molecules that we're talking about, they're covalently bonded, and they all have carbons, so that means that they're organic molecules. And we link them together by dehydration synthesis. Our examples of our disaccharides are milk sugar, it would be lactose, maltose is malt sugar, and sucrose is the sugar that we buy in the store, table sugar. So our polysaccharides, what we do to make polysaccharides are we just link a bunch of glucose molecules together. We link a bunch of monosaccharides together through dehydration synthesis, and we get our complex carbohydrates that we call polysaccharides. Polysaccharides, unlike the monosaccharides and disaccharides, are insoluble. We usually cannot digest them. We can't digest a lot of them. The ones that we call complex carbohydrates that we can't digest, we call them fiber. The ones that we can digest some of them, like in the ones in potatoes, those are starch. Cellulose is definitely fiber. Those make up the cell walls of plants. Now glycogen is a storage form of polysaccharides in animals. We store them in our liver cells and our skeletal muscles. Remember, we just link glucose molecules together when the pancreas releases a certain hormone. I want you to pause this video and renew your notes from the first day of class. We went over that hormone. All right, here are pictures of monosaccharides. We've got glucose, fructose, and galactose. These guys are structural isomers of one another. They all have the same formula, C6H12O6. And then we have deoxyribose and ribose. These are our monosaccharides. This one is in DNA, and this one is in RNA. Down here we have our disaccharides, sucrose, maltose, and lactose. We just add two monosaccharides together to get these guys. Two monosaccharides, two monosaccharides. These are easily digested when we ingest these. When we build them through dehydration synthesis. When we break them down using hydrolysis. Now this is what glycogen looks like. It's branching structure, and each one of these green things, a glycogen represents a glucose molecule linked. That is a little covalent bond between each glucose molecule. So this would be what we would find inside of your liver cells. And if we break these covalent bonds, the liver cell can release one of these little glucose molecules right into the blood to raise your blood glucose levels. This is what cellulose looks like. This is what cellulose looks like. Cellulose is just repeating glucose units, but we can't break cellulose down because we don't have the enzymes to do that. So this is fiber. When we eat cells of plants, this fiber goes through our gut and goes right out the other end. We call it fiber. So we can't digest it. It helps. It's roughage. Okay, that's the end of this section.